about mitosis and certain genes which regulate the different steps during the cell cycle, right? And what is the role of such genes in development of cancer? So let's start with a very simple concept that what is cell cycle, right? Cell cycle is basically a concept related with that how a cell passes through different phases to divide into two daughter cells, right? What is cell cycle? Cell cycle is consisting of all those events and phases through which a cell passes, a mature cell passes eventually to divide into two cells, right? Now let's start that normally cell cycle has two parts. Number one is called interphase and second is called mitosis. So let's go with the examples. Let's suppose there is a cell here, right? And this cell plans to divide, right? This is a mature cell here and it gets stimulus and it is going to divide. Of course, when cell is going to divide, it has to duplicate its genetic material. It's going to pass through the mitotic division. So it has to duplicate its genetic material as well as it has to increase its what? Cytoplasm, proteins and organelles. Now, when this cell is planning to go for division, now, first of all, it has to duplicate its DNA. Is that right? So, let's suppose cell in this stage, it is duplicating its DNA and I put DNA molecule which is open and it is being replicated. It is being replicated over here, DNA molecule. Now, when cell plan to undergo division, it does not directly start replicating its DNA. Before starting the replicating its DNA, cell has to produce lot of proteins and lot of enzymes, right? Increase its cytoplasm, increase its organelles and produce those enzymes, right, which can help in replication of DNA and produce all those proteins which should help in replication of DNA. So this stage where DNA is being synthesized, we call it S stage. S phase is phase of DNA synthesis and phase of cell before the DNA synthesis, right? That is called pre-synthetic phase. What is this phase? Pre-synthetic phase. Synthetic means synthesis of DNA. Now this phase, which is pre-synthetic phase, right? This is also called gap 1 or growth phase 1 or simply we call it G1. Is that right? So when a cell plans to divide, first it will enter into G1 phase where it will do all its preparations at molecular level and organelle level to synthesize, to get ready to synthesize the DNA. Then DNA synthesis process will start, right? So during that time when DNA synthesis is going, we call this phase S phase. And once the DNA has been synthesized, means DNA has been semi-conservatively replicated. When DNA has been replicated, right, it means that 2N has become 4N, right? Or we can say when DNA is replicated, every chromosome has become double structured chromosome, right? After that, once DNA replication is complete, now it has to go to next phase. In the next phase, it should get ready. It should get ready to divide that genetic material which has been duplicated for the two daughter cells. Now, when cell is preparing, after the synthesis, when cell is preparing itself to divide the nuclear genetic material equally into two daughter nuclei, right, that preparation phase is called G2 or growth phase 2 or gap 2. So what is G2? G2 is post-synthetic but pre-mitotic phase. Post-synthetic but pre-mitotic phase. During G2 phase, what's going on? Cell is getting ready to divide its duplicated DNA into two daughter nuclei. Is that right? Now, once it has been through the G2 phase eventually it will enter into next phase where it is going to divide the genetic material, and this nuclear membrane will eventually dissolve and genetic material will go to both sides of cell so that it can go for the new two daughter nuclei. Now that phase in which nuclear duplicated or replicated DNA or chromosomes, they are under the process of division, is that right? For the two daughter nuclei, this stage is called mitotic stage. What is it? Mitosis stage, or we simply call it M phase, right? Now, after the M phase, eventually when cell is divided into two cells, right? It is divided into two cells. In some cases, in some tissues, cell directly enter into next cell division. In some other tissues, cell may go and take rest, right? And if cell goes out of the replicating cycle or proliferative cycle, we say it has gone to growth phase, which is G0. Is that right? So let me repeat it rapidly. Rather, why don't you repeat for me? Yes. Cell cycle, what is cell cycle? Cell cycle means all the events which are occurring in a cell when it is going to divide from one cell into two cells. To divide one cell into two cells, we have to basically make cytoplasm more, organelles more, and especially DNA should be replicated and equally divided into two daughter cells. Now, first G1 phase, what is that? Cell is getting ready for synthesis of DNA. It is getting ready for synthesis of DNA. It is not yet dividing its DNA, right? So it is pre-synthetic phase, is that right? Then DNA replication start or synthesis start, right? S phase. Then cell enter into the preparation that how replicated DNA should be divided for two cells. For example, here it will make the proteins which are related with, yes, spindle formation. Once those proteins which are required for division of cell, they are made, then cell will start separating its chromatids or replicated DNA and go through processes which will make two daughter nuclei and eventually two daughter cells. That is mitosis, right? And as soon as it ends up, mitosis end up, in some tissues cell may go again into proliferation, and in other tissues cell may exit out of the cycle and start resting, and they may come into cycle again if there is appropriate stimulus for proliferation, right? Now, those tissues in which cells are cycling continuously, there are some tissues in our body that cells there are continuously multiplying. It means as soon as cell completes its mitosis, it enters into G1. Cell does not go to G0, and G1 and again process start. Can you tell me some tissues like that? Yes, please. Yes, please. Skin, very good. Skin cells, right? They are all the time multiplying. Any other tissue? Gastrointestinal cells, they are all the time multiplying. Is that right? So such cells or tissues are called, that in these tissues where cells are multiplying all the time, we call those tissues, the cells there are labile cells. Labile cells are those cells which are multiplying all the time in the tissues, and these cells, once they complete mitosis, they don't enter in G0. Once they complete one division, they go for the next division. Is that right? They are labile cells. Such cells are seen classically in your gastrointestinal system. Such cells are also seen in the skin. Some of the skin cells are continuously replicating. They don't go into G0 phase. And then such cells are also present in bone marrow, hematopoietic cells. Hematopoietic cells also keep on multiplying. After the labile cells, then there are some other tissues in which once cells have completed a cycle, if that tissue does not require more cells will go into G0 stage, and cells will remain in G0 stage, where they are doing housekeeping function. The genes which are concerned with housekeeping of the cell, only those genes are working, plus if that tissue has some special function, those genes are working, but cell is not planning to divide. So in some tissues, cells come out of the cycle, go into G0, but with appropriate stimulus, they can be forced to proliferate again, right? Such tissues are said to be stable tissues. Stable tissues mean that, or stable cell, in some tissues there are stable cells. Stable cells are those cells when in the tissue most of the cells are in which phase? G0, but normally they are not regularly multiplying, but if there is appropriate stimulus, they can multiply. Is that right? So can you tell me example of stable tissue? Yes, please. Not neuronal cell, not muscle cell, you are going to miss your MCQ. Yes, stable tissues where cells are normally not replicating, but if there is appropriate stimulus, cell can leave the G1 stage and enter into this. Rapido. No, none of you knows it. Wow. Why don't you tell me liver, hepatocytes? Hepatocytes are normally not multiplying in a very fast rate. Most of the hepatocytes, most of the time are in G0, but if you cut a piece of liver, then cells will come out of G0, they will enter into cycle G1, and then they will start multiplying and make it good. Is that right? Cells in the kidney, tubules, nephron cells, cells in the pancreas. So these cells, which are not in these cells, in these tissues, there are stable cells. What are stable cell population? Where most of the cells are arrested in G0 and they are just doing the general and special functions but not planning to multiply. But if there is appropriate stimulus, they can be brought into cycle and multiply. Clear? Then there are some tissues where cells are permanent cells. Permanent cells mean that in those tissues, once cells have divided, they go into G0 phase, but they cannot re-enter into G1. In some tissues, some cells, or most of the cells, once they have multiplied and gone into G0 phase, right, you cannot bring them back into cycle. So in those tissues, cells are permanent and they don't multiply. If you want to increase the cells in those tissues, then you have to find the stem cells in those tissues which should multiply. But in these tissues where there are permanent cells, once the permanent cells are well differentiated, they are doing their general and specific function, they cannot be pulled back into cycle. Yes, what are these tissues? What are these cells? Now you can tell me neuronal cells, neurons, they are permanent. Is that right? You cannot drag the neurons back into multiplication or proliferation cycle. Then there are muscle cells. Don't tell me just muscle cells. Smooth muscle can proliferate. Tell me skeletal muscle cells and myocardial cells. Right? So skeletal muscle cells and myocardial cells, they are permanent cells that once they are fully differentiated, you cannot, they will go into G0 phase and you cannot drag them back over here. Am I clear? Any problem up to this? Now, one thing which is very important to understand, first of all, I will go briefly into stages of mitosis. How do you define mitosis? Yes, mitosis.
Division of cells.
Division, she thinks division of cell is mitosis. Anyone will come with better definition? What is mitosis? Let me tell you. Don't confuse. First you should be clear what is in cell cycle, what is mitosis and what is interphase. Interphase is G1 phase plus what is this? S phase and plus G2 phase. It means when cell is planning and making lot of proteins so that it can enter into DNA synthesis, plus when cell is replicating its DNA, and plus when cell is making proteins and organelles ready for mitosis, we say all these stages from here, G1, S up to G2, all these stages, right, they are together called interphase. What are they called? Interphase. But this stage is called mitosis. Now I want that you should be very clear what is mitosis. Yes.
Somatic cells.
Okay, he is telling us somatic cells when they divide by mitosis, you are right, and germ cell, when they divide, eventually they go through meiotic division. I'm asking, what is mitosis? Yes, please.
One cell gets together cells that are identical to each other in the identical cell.
More specifically, the nucleus.
It is about the division of nuclear material. Listen carefully. When I say there is mitosis going on, what does it mean? That is at this stage. During the process of mitosis, right, the replicated or duplicated genetic material in one nucleus should be divided for two daughter nuclei. So when you are talking about mitosis, we are specifically, yes, focusing on division of nuclear material. Is that right? That cell during the synthesis has duplicated its genetic material. And when cells start the process of dividing the duplicated genetic material or replicated genetic material into two sets of identical genetic material for two daughter cells, that is mitosis. And the process in which cytoplasm and organelles also divide into two and cell membrane separate into two, that cytoplasmic and its organelles division into two cells is called cytokinesis. So you should be clear about mitosis and cytokinesis. What is mitosis? Mitosis is that once synthesis of DNA has been done, once DNA has been replicated, after that, when it is going to duplicated DNA or duplicated genetic material of the nucleus is going to under process so that it can divide the nuclear material duplicated for the daughter cells. This is mitosis. And during this process, mitosis is going for the nuclear material. But during the mitosis, also organelles and cytoplasm and cell membranes are undergoing division, so that not only there are two daughter nuclei, but there are two daughter cells. And this process of dividing the cell into two daughter cells is called cytoplasm of the cell. When divided into two, we call it cytokinesis. Is that right? Any problem here? No. Now, very briefly, what are the stages of mitosis? I know all of you know it very well. They start teaching from high school and they keep on teaching doctors until they are there. So stages of mitosis, before I go into really detail, attention please, again, never ever in your life mix the mitosis with the interphase. Interphase is when cell is planning to divide, getting ready for division. Is that right? That includes G1 pre-synthetic phase, synthetic phase, pre-mitotic phase, right? This is all interphase, and this is, I'm going to explain phases of mitosis, in which mainly we are focusing on what? Nuclear material division. Is that right? Now, this nuclear division during the mitosis, there are four phases of mitosis. What are those four phases of mitosis? Who is going to tell me? Four phases of mitosis. I know, everyone knows that very well. Yes. Yes, she knows it's prophase. Very good. Then it will go into what? Prophase into metaphase. And then, okay, let me put it prophase here. Prophase goes into, yes, metaphase, and metaphase goes into anaphase, and anaphase goes into telophase. Is that right? How do you remember that? You remember them or not? You remember them. That's good. But only there's an easy way to remember that. Just P and mate. Right? P, prophase, metaphase, anaphase, telophase. Is that right? Okay. Don't stimulate your imagination too much. Concentrate on the lecture. Now, what happens in the prophase? Listen, this prophase which I'm drawing here, actually, if I say that this is G2 and mitosis is going from here up to here. Is that right? Now, when cell is going through this phase, what are the changes coming? Of course, you know the changes are prophase, yes, metaphase, anaphase, and telophase. But this is not enough to know. What happens during prophase? Let me make a cell here, right? And we are going to see what happens in this cell during the prophase. Now, just before the prophase, cell was going through G2. During G2, what has happened? This is a nucleus, nuclear membrane. At this phase I'm talking about, before just prophase, right? There is what? G2 phase. There is well clear cut. What is this? Nuclear membrane. The DNA is prominent or not during G2? Chromosomes are well prominent or not? Once the normally when DNA replicates itself, it opens up, right? And it condenses during the prophase. So originally in the beginning of before the prophase, DNA is very light. Okay, let me give you some different color for DNA. DNA is double structured, but genetic material across, they are making loose network. This is before the prophase. And centrioles may be randomly present over there. Now this cell which is in G2, what is it? This cell is in G2. Now it is about to enter, or it is entering into prophase. When it is entering into prophase, I told you it is phase of mitosis, and mitosis means separation of genetic material of nucleus into two identical sets. So what should happen? Logically tell me, what should be the next step in this cell? What should be the next step? First of all, before you do anything, these loose chromatin condense itself. What should happen? This loose chromatin condense itself. When loose chromatin will condense itself, then what will happen? That chromosomes can be seen more clearly because they become condensed. And every chromosome will be single structure or double structured? It will be double structure because during synthesis phase we replicated the DNA. So now here where chromatin is loose, it becomes condensed chromatin, and you are able to see the chromosomes double structure. Is that right? So what is the first step in prophase? Chromatin become condensed, and what is becoming more visible? Chromosomes which are visible as double structured. Plus, do you think if nuclear membrane remain intact, can you divide this into two daughter nuclei? No. So next step is that nuclear membrane start dissolving. Nuclear membrane start dissolving. And with that, we are going to make spindle, and to make a proper spindle, we have to get this centrioles at the, what is this? At the edges. Two opposite pole. So these are the three basic changes which occur during prophase. That chromatin become condensed, number one. Number two, nuclear membrane become indistinct and start dissolving and disappearing, because there are special nuclear proteins here which are called laminin, and some enzymes will come and they will phosphorylate the laminin proteins, and when laminin protein becomes phosphorylated, nuclear membrane start dissolving. In the same way, in the chromosome there are proteins called histones, histone 1. When histone 1 gets phosphorylated, the nucleus these chromosomes start getting condensed. Is that right? So phosphorylation of histones in, what is this, chromatin condenses this, phosphorylation of laminin in the nuclear membrane dissolve this, and arrangement of centrioles at the polar end. This is prophase. As soon as, now listen carefully, as soon as this membrane totally disappear and microtubules start developing clearly, cell will go into next phase. Let's suppose this is here. Now nuclear membrane has disappeared. Centrioles are present over here. This is one centriole, this is other centriole, and tubules start extending. right? Microtubules. Why this spindle is being made? Because these double structured chromosomes should be arranged on that structure before the division. Is that right? So that there should not be mistake during the division of genetic material. So next phase is nuclear membrane has been disappeared and microtubules form, mitotic spindle is formed, and your genetic material, which now consists of what? Chromosome which are double structured. They are arranged on equatorial plane, right? And when mitotic spindle is very clearly formed and dense chromosomes are arranged on the spindle, and especially these, what is this, tubular microtubule get attached. What is this point? Kinetochores. The point where two chromatid, what are chromatid? When one chromosome has double structure, each structure is called chromatid. Both structures are identical. Chromatids, look, this is single structured chromosome, and after the S phase, every chromosome becomes double structured. And this double structured chromosome has two identical, right, material. Each material is called chromatid 1 and chromatid 2. Now each chromosome consists of two chromatid because each chromatid will go to one daughter cell. And plus, this is a special point where the tubule will bind